so today we're going to do a very basic introduction to the structure of the brain. It will be quite simple, just enough background so that you can follow along in this course if you don't already have any neuroscience background. So now, even if you perhaps don't know much about the brain, you probably know where the brain is. So the brain is, of course, in the head. And your head is overall a very impressive control center for your body. So in addition to the brain, your head houses many of your sensory organs. So your brain has obviously your eyes, your ears, your taste organ, your tongue, and much of the role of the brain is to process the signals that are collected by these sensory organs. And so the sensory organs are connected to the brain. Most of them are connected to the brain through the thalamus. So for example, from your ear, the signal goes to this subcortical structure called the thalamus. It's sort of in the middle of the head, and then relays the signal to the relevant sensory cortex. Similarly, for the tongue and for the eye, the signals go through the thalamus to the sensory cortex. So for example, from the eye to the visual cortex. Interestingly, from your nose, the signal goes straight to the smell cortex, the olfactory cortex at the very front of your brain. So that pathway is a little bit different. But that's kind of how the different sensory pathways are organized inside your head. So now here we want to talk about the basic physiology of the brain. And the reason we want to understand it is that that understanding will constrain our hypothesis spaces about brain function. So maybe different parts of the brain perform different functions. That's a possibility. And most importantly, when we develop theories about the neurobiology of cognition, those theories need to make sense given the physiology of the brain. So we don't, for example, want to postulate that signal goes from area A to area B if those regions actually are not physiologically connected. So we need to have some understanding of the basic biology of the brain in order to do neurobiology of cognition. It should be kind of obvious. So now when we start looking at the brain, the first observation usually is that you notice that the brain is a very, very wrinkled organ. It looks kind of funny. And it's wrinkled in a way that's consistent between individuals. And so here, for example, I'm pointing to some big wrinkles that you'll observe in most brains. And given that the wrinkledness of the brain is such an important property of it, we have terminology to talk about those wrinkles. So gyri, gyrus as the singular, those are the convex parts of the wrinkles. And sulci, or sulcus as the singular, those are the concave parts of the wrinkles. And when you have a really prominent sulcus, that's often called a fissure. Okay, so some sulci are really deep, and we call them fissures a lot of the time. And in fact, there is one really deep fissure right in the middle of the brain. It's called the medial longitudinal fissure that divides the brain into the two halves that you all know about. So your left hemisphere and the right hemisphere. So inside your brain, there's kind of like these two brains sitting right next to each other. And they are connected. So your left and right brains talk to each other. And much of that is through a prominent fiber tract that's called the corpus callosum. So that's the structure that connects the two hemispheres and allow them to talk to each other. So now within the hemispheres, there's also a consistent structure. So each hemisphere divides into 4 subsections, and those subsections are what we call the lobes. And so here you see the 4 lobes. So we have the frontal lobe, we have the parietal lobe, we have the temporal lobe, and the occipital lobe. And here are some of the major fissures that then divide the or separate the lobes from each other. So for example, the lateral fissure, or often called Sylvian fissure, is a major lobe divider right here. So separates the frontal lobe from the temporal lobe from the parietal lobe, and so forth. And here we also have this central fissure that divides or separates the frontal lobe from the parietal lobe. And then within the lobes, the structure of gyri and sulci is also quite consistent across individuals. And here you see some of the terminology for talking about the different gyri and sulci. And also most of it makes a lot of intuitive sense. So for example, let's look at here in the temporal lobe. The bottommost gyrus is the inferior meaning on the bottom, inferior temporal gyrus, and then we have the middle temporal gyrus, and then the superior temporal gyrus on top. The same organization repeats itself in the frontal cortex. So again, we have the inferior frontal gyrus, middle frontal gyrus, superior frontal gyrus. And then there's also other nomenclatures for talking about the different structures. So here, for example, angular gyrus, supramarginal gyrus, structures of the parietal cortex, also language relevant. So we will hear about those regions in this course. Okay, so that's sort of at the, at a very broad level. So what is the brain then made out of? What kinds of cells do we have in our brains? So brain cells, of course, are called neurons or nerve cells. And so here we have a picture of a neuron. Our central nervous system is made up of about 86 billion neurons, 85, 86 billion neurons. That number changes as our understanding evolves. So you'll find older textbooks that will cite a different number. And unlike cells, unlike other types of cells, nerve cells are special in that they can both receive and send out signals from neighboring neurons in the form of electrical pulses. So your brain activity is, in essence, electrical activity, with electricity rushing through the neurons in order to allow them to communicate. And typically, any given neuron is connected to about 10,000 other neurons. And the specific point of contact between what's called the axon, so this is a long part of the neuron that transmits the signal from one place to another. So the contact point between the axon and the next nerve cell is what we call a synapse. So here's a picture of a nerve cell. We have a cell body, then we have this long structure axon, and then we see the next nerve cell over here, and then this kind of hairy part of the nerve cell that receives the signal from the other neuron is what's called a dendrite. So over here we can see the dendrites of this nerve cell, and then that nerve cell makes contact with the nerve cell of another a dendrite of another neuron. Let's watch this quick clip that also explains.
Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
That gives you a basic idea of the structure of neurons and what they do in your body. So understanding the structure of neurons is also key to understanding the difference between gray matter and white matter. Again, terms that you probably have encountered before. So gray matter, that's the cortex. That's sort of the skin of your brain. It's the part of your brain that houses the majority of the computational power of this organ. And the gray matter consists of the cell bodies and the dendrites. So that's, basically what the gray matter is. And the white matter, so that's the inner matter, is then consists of these myelinated axons. So that's the tissue through which these messages pass between different areas of the gray matter. Here's another visualization of that. So here you have your gray matter. So that's the cortex housing the dendrites, synapses, and then we have the white matter. So here you can see an example axon passing from one part of the cortex to another. So now the cortex further has a complex structure of different types of nerve cells. So I haven't gotten into the different types of nerve cells in detail, but there are different types of neurons and they're arranged into layers in your cortex. And not all parts of the cortex are the same when it comes to these layers. So the layering structure differs between different regions. And the arrangement of nerve cells in the cortex is what's called cytoarchitectonics. So when we study cytoarchitectonics, we're studying the arrangement of nerve cells in the cortex. So there can be up to 6 layers of different types of nerve cells in your cortex. And this has some sort of pragmatic importance because a German neurologist, Brodmann, studied the cytoarchitectonics of the cortex in great detail in the 19th century, and he, on the basis of this study, defined the cerebral cortex into 52 distinct regions on the basis of the cellular structure of the different regions. And this division is actually still something, this particular parcellation that Brodmann developed is still broadly used even today. And so here he just gave all the regions numbers. And so all these, here I have all the Brodmann areas numbered. And we use these numbers quite commonly to refer to different parts of the brain. So now here, there's no reason for you to learn these numbers by heart. You can always refer to the map. And so many of my students, for example, have a map of the Brodmann regions next to their desks. And so you can kind of have a quick reference point. There are many different types of parcellations as well, but this is one of the oldest ones and one that really has stood the test of time. So let's just do a quick review of what we talked about so far. So we're talking about the brain. The brain divides into 2 hemispheres. Within the hemispheres, we have 4 lobes. in each hemisphere, and within the lobes there's a relatively consistent structure of gyri and sulci, so that's a consistent wrinkling within the brain. Your brain is made out of neurons, so that's the cell type that we have in our brains. The brain can also be divided into gray matter, or the cortex, versus white matter. Gray matter houses the cell bodies and dendrites, and white matter houses the axons. And your cortex then further has different layers, so that's the arrangement of different types of cell types within the cortex. And on the basis of that arrangement, we refer to different parts of the brain in terms of Brodmann areas. And so that's the parcellation that Corbinian Brodmann developed. Okay, this little video is not about brain function. I'm not getting into that in any detail here, but I'm just going to give you some super broad generalizations that you'll find, in sort of introductory textbooks. So the frontal lobe is generally thought of as the sort of main executive center of your brain. Participates in decision making, problem solving, higher level cognition like that. The temporal lobe, very important for language. We'll talk about the temporal lobe a lot in this course. Also important for memory, important for audition. That's your auditory cortex is in the temporal lobe. Parietal lobe is kind of more complicated. We will more complicated to make broad generalizations about, but it does participate in language processing, so we will encounter the parietal lobe in this course. And then the occipital lobe, of course, is your main vision center. So your visual cortex is in the occipital lobe, and so that's sort of thought to be the this lobe's function at large, is to do vision. So now, since we'll be talking about the brain, I just want to introduce a little bit more terminology. So this is a picture that just shows us how we kind of talk about directionality within the brain, and it's really the same as for any biological system. So when, for example, some type of activity is relatively towards the front of the brain, then we can use the adjective anterior for it, or rostral, which comes more from the animal literature, but you'll notice that term as well. And then the opposite of that is posterior or caudal. Again, that's the more kind of animal literature linked term. And then when we're talking about bottom versus top, then it's either inferior versus superior, or ventral versus dorsal. And so those are, again, sort of equivalent. You'll also see different ways to describe the kind of slice that we're taking through the brain in order to visualize activity. So you can take a coronal slice. Here you can see sort of like a slice through the middle. Horizontal plane, so that's the horizontal type of slice. And then sagittal is what we describe a slice that goes through the middle this way. Better way to explain it. And then finally here, this is just what we already talked about, superior versus inferior, but then also lateral versus medial. So that's actually something that you'll really need to understand. And so when it's more lateral, then it's more towards the side. When it's more medial, it's more towards the center. And of course, the brain also has the medial cortex. So both of the hemispheres have a lot of cortex that's right in the middle of the brain. And so that's what we call the medial wall. And it will not be irrelevant for what we'll be talking about in this class. Finally, if activity happens on both sides of the brain, we call it bilateral. If it's on the same side of the brain as something else, so let's just say like on the same side as the relevant sensory organ, then it would be ipsilateral. Contralateral refers to the opposite side of the brain. So for example, if I'm delivering an auditory stimulus to the right ear and I'm interested in the response of the left hemisphere to that stimulus, then I would be talking about the contralateral response to that auditory stimulus. So that's it for today. And next, we will be taking a peek into the history of the field, mostly focusing on what we've learned from aphasiology as regards to the brain basis of language. See you then.